Dear participants, I welcome you all to this 1 week webinar come FDP series. Today we have a special speaker and I take privilege in introducing Professor Geetha K. Vemuganti. Ma'am has done her MD from NIMS Hyderabad in 1993. She was the founder dean, School of Medical Sciences, University of Hyderabad till November 2017. Professor Geetha was the head of LV Prasad Eye Institute till 2010, and she's a consultant pathologist at Medwin Hospitals, Hyderabad. Professor Geetha is also adjunct faculty, University of Rochester, US, from 2009 to 2013. Well, ma'am has been an observer at Center for Disease Control, Atlanta, GA, USA, and she has been invited as visiting pathologist to Eye and Ear Infirmary, University of Illinois, Chicago, USA. Well, ma'am's teaching experience include she's a recognized guide to PhD students at BITS Pilani UOH, that's University of Hyderabad, and talking about publications, the number is 232. Well, that's immense contribution to the society, ma'am. Your research is commendable. She has papers of about 186, abstracts 33, book chapter 13, and ma'am's citation index is as high as 5,557 and H index of 43. Well, Professor Geetha has attended more than 300 national meetings and 100 international meetings. Well, talking about awards and recognitions, well, the list can go on and on, but because of the time constraint, I will reduce this. Ma'am is a member of ICMR expert group on stem cell research and therapy since August 2018. She's the elected fellow of Telangana State Academy of Sciences since Jan 2018. Ma'am has received Visishta Puraskar 2017 by Dr. Ramineni Foundation, USA. Senior Achievement Award from American Academy of Ophthalmology. Well, that's a big award, ma'am. Congratulations. And she's a member of National Apex Committee, Stem Cell Research and Therapy. And ma'am has been awarded 5 best photographs by the Ophthalmic Photographic Society at the American Society of Cataract and Refractory Surgery, San Diego. Ma'am has received grants from reputed funding agencies. And ma'am, thank you very much. It's an honor to have you on this webinar series, ma'am. Thank you for accepting our invite and coming over to share your profound knowledge with the participants across the world. Thank you very much, ma'am.
Good morning, students. It's a pleasure to talk on a topic that is of great interest to me. And I understand many of you are engineering students. So let's see if we can find a common ground to discuss our common topic of tissue engineering. So I'm Geetha Vemuganti from School of Medical Sciences, University of Hyderabad. How would you approach tissue engineering? What is tissue engineering? So these are the things I think I would like both all of us to be on the same page. The questions that we should ask is, what tissues or organs are we going to make? What do we need to learn before we start this? What materials do we require? What are the regulatory approvals required to proceed with these studies? How do we understand the structure function relationship, and how do we provide proof of concept before we go ahead with what is called as a hierarchy of research, which starts from basic research, it goes to preclinical, then clinical research, and then finally it goes to translational research. So the organization of the human body Being biomedical engineering students, I guess, and I hope you would have gone through the structure and function of the human body, like atoms or the electrons, which are the smallest units. In human body, the cells are the smallest functional units. Group of cells make tissues, and a group of different kinds of cells make tissues, and these different kinds of cells have matrices which bind them together so that the cells can talk to each other. Example, epithelium, bone, cartilage, that's a tissue. And different kinds of tissue make an organ, and an organ is a functional unit of a system. Example, heart of a cardiovascular system, kidney in urogenital system. Different kinds of organs make a system, that is the gastrointestinal tract or cardiovascular system, central nervous system, and so on and so forth. And different systems make a human body. I hope we are all familiar with this. And this is how we would, this picture is from the Wikipedia. The human body can be divided in various ways. It can be a regional anatomy where we talk about head and neck, brain, thorax, abdomen, upper limb, lower limb, or we can talk about the systems that I mentioned to you about. Integumentary, that is the skin, the skeletal muscle system, the skeletal system, muscular system, nervous system, cardiovascular, lymphatic, endocrine, digestive, hematopoietic, and so on. Respiratory, urogenital. So each of these systems will have different organs. However, remember that the cells in these different kinds of organs and different tissues are similar. They can be epithelial cells in all these tissues, connective tissue in all these tissues. We will learn as we go by. Now, how are these different tissues formed in human body? It's a miracle that in 9 months, from one cell, an entire human being is formed. And as you know, the intrauterine life is divided into three: first trimester, second trimester, and third trimester. And within the first 3 months of intrauterine life, from one cell, the entire fetus with all the designated organs are formed. In the second trimester, they keep growing, and in the final trimester, they attain the full size and function and are ready to come to extrauterine life. And this is the scale in which the different organs are formed in the intrauterine life. So, from one cell, the cells keep proliferating, differentiating into different organs. So, what we are trying to do in tissue engineering is mimic these changes which occur in the intrauterine. This is, we can hardly mimic it, but still we can make an attempt using different tools. Now, what is engineering? You all know, you have to train me for that. So, engineering is the science, it's a skill and a profession of acquiring and applying scientific, economic, social, practical knowledge in order to design and also build structures, machines, devices, systems, materials, and processes. So, here we would try to attempt to build human tissues with the combined knowledge that we all have. So if you have to make a sketch, or if you may want to make a car, Tesla car, or if you want to make a boat, what do you do? The same principles that we are all being taught: ask, acquire, assess, analyze, apply, and audit principles. So basically, in tissue engineering, we need to now look at the tissues, how they are formed, what they are. So what do we need to learn now? We have asked the question that we need to make a particular tissue. Now, what do we learn? What do we um, review? So it's nothing new. From Hippocrates' time, he's a Greek physician, father of medicine, and followed by Herophilus and Vesalius. These are the giants who have taught us what the human body is about, how different kinds of, they would even steal the bodies from graves so that they could understand the anatomy better. So, anatomy is the most important thing when we understand the tissues. Then we also have to understand how they are structured, that is the histology. So I would compare creating a tissue like how you would create a home or a building. So what cells do you need? Where do you get them from? What is the matrix or what is the cement that you would use to bind them? What are the factors that you would add to grow them? What is the purpose? So, just like you would make a building based on the need, are you building a church? Are you building a temple or a home, a hut, a gymnasium? So, it all depends on what is the purpose for which you're making. So, even though you're an engineering student, you have to know the purpose for which you're making the tissues. So this sequence that we continuously always talk about, like atoms, molecules, organelles, cell, tissue, organ. So this is a hierarchy or organization that we have to keep in mind. Unlike a building, here the cells, people work it at home, but the tissues have to do their own work themselves. They have to talk to each other, they have to carry on. So, bottom you have a cell which makes an epithelium, and these cells talk to each other through intercellular bridges. And below that is the matrix or the soft tissues or the connective tissue which bind them together. And then they form the organ. Another way of looking at the body is forget about the systems, forget about different organs. Look at the different types of cells which are capable of growing or repairing. So, based on the repair capacity of the cells, all the cells in the body from head to foot can be divided into three types of cells. These can be called as labile cells. That means they continue to divide throughout our lifetime. We are all alive because our bone marrow continues to produce a lot of RBCs, WBCs, and platelets so that we are healthy. The skin continues to grow, so therefore we don't have any patches of areas where we don't have skin. Our mouth doesn't have ulcers because it's healing by itself. So these are the tissues which are called labile cells. They heal by themselves, and that's because they have a population of what is called as stem cell population. The second type of cells are called stable cells. That means they come to our rescue as and when required, SOS. And these are the liver cells, the bone cells, the mesenchyme. If you have a fracture, always, even if you don't do anything, the bone will heal, except, of course, the alignment. If it's not proper, it will not align properly. So these stable cells in different organs have limited scope of regeneration and repair. They require a stimulus, unlike the labile cells, where the bone marrow doesn't ask you, should I produce more RBCs for you? The third category of cells are the permanent cells. We believed that they do not have stem cells, but however, recent evidence points out that there are stem cells even in these permanent tissues, and these include central nervous system, skeletal muscles. When you go to the gym, you have wonderful expanded muscles and thick biceps, triceps, but after 6 months, if you don't go, they will reduce in size. And the reason is because the skeletal muscle fibers have become enlarged, but the number of cells remain the same. Similarly, cardiac muscle, glomerulus, if these structures are damaged, there is practically no scope of regeneration. Research, however, points out that there could potentially be a possibility of repair, but however, it is still in a research phase and it is not adequate to compensate for any damage which happens in clinical conditions. Now, how does this division, how do they divide? As I'm sure you all have read about the cell cycle. When I said the stem cells, I will come to it a little later. So all the dividing cells go through what is called as a cell cycle. And in this cycle, there are two gap G1 and G0, G1 and G2, and then there are two intervening phases of mitosis. So basically, when the cell is resting, it is called G0. Then it goes through a growth or a gap phase where the cellular contents are increased, they are duplicated, and they prepare for what is called as a synthetic phase. And in the S phase, these chromosomes are duplicated within the cell. And then it takes again a gap or a growth phase where the double checks, it took the duplicated chromosomes for errors and any other repairs, and then it goes to a phase of mitosis. And in mitosis, as you're already familiar with, There are different phases of mitosis: prophase, metaphase, anaphase, telophase, and this is carried on by different growth factors, by cytokine release, and so on. So a cell which is a labile cell is always in a cell cycle. The stable cell is in G0. When required, it goes into the cell cycle. In permanent cells, they are always in G0. They may not enter this cell cycle again, and that is why there is no repair or no regeneration. So this is the link between the types of cell and the cell cycle. Now, what are the stem cells? These are also dividing cells. They can divide, but they have another unique property. By themselves, they are undifferentiated, but they are capable not just of differentiation, but a unique quality called as a self-renewal. What does it mean? You see on the right side, there is a diagram. There is one cell in a niche, in a cavity which is there, and the surrounding is the niche. So if that is a stem cell, it will divide into two daughter cells. One goes back to the stem cell compartment, whereas the others are pushed into differentiation into further down the lineage. Whether this niche is all or none phenomena, is a gradual phenomena, is an anatomic structure or functional structure, we are trying to understand this. Um, uh, there are some physicists and mathematicians who try to evaluate how come one daughter cell goes into self-renewal and one daughter cell goes into differentiation. They're trying to even have a mathematical modeling for that, and some of you may probably address that. So the challenge lies in identifying these stem cells, isolating them, and characterizing. So why cells are not enough? Why do we need stem cells? As I mentioned before, if they don't divide, if some cells don't divide, we need to look at stem cells for tissue engineering. And what are the different types of stem cells? We have embryonic. So this is the hierarchy as we talked about in the first slide. From one single embryo, we form different types of parts in the body. So here we have these cells can be divided as embryonic or embryonic stem cells or embryonic germ cells, fetal cells, adult cells. So the difference between any dividing cell and stem cell is the unique property of self-renewal. And the different types of adult stem cells make about 0.5 to 5% of all cells. And these unique properties of stem cells are because they have an asymmetric division and they have low mitotic cavity, they have error-free proliferation, and by themselves they are poorly differentiated. And you would think that these stem cells will divide and divide. No, actually they are quiescent. They will divide at their own time and for homeostasis. And they are always protected in a special anatomic niche, and I call them under bunker army. These properties are endowed on these stem cells, both by intrinsic properties as well as the niche-driven factors, that is the blood vessels, the special stroma which is around it. So various types of adult stem cells are there, as we talked about, in the skin, in the intestines, in the mammary glands, and in different parts of the kidney as well, in the tubular structures of the kidney, not the glomerulus. So coming back, if we need tissues to be regenerated or to be reconstructed, we have to understand how the cells, what are the different types of tissues and how they work together. So the types of tissues are epithelial. All the ones which are lining, whether it is the skin or the mucus, are called epithelial. Connective tissue, muscle, nerves. So when you talk about tissue engineering, which cells are we looking at? So we need to understand that. So these are the different types of blood is a connective tissue. Do we want to make blood? Do you want to make endometrium? Do you want to make a blood vessel? Do you want to make a heart muscle? So we need to specify what we need to make. And for that, unless you understand the histology, the tissue structure, different types of cells, we cannot make progress in tissue engineering. So the reason why the epithelial cells are special are because of the function. Now we talk about the structure-function correlation. If we need to make a tissue, obviously it should serve the purpose. So if you're making an epithelium, it should serve the purpose of protection, absorption, wherever the tissue is. that particular function is assigned to the epithelium. So, therefore, we need to understand the basic structure function correlation. If you want to make a connective tissue like blood, skeletal muscle, so these are the different kinds of connective tissue, that bone, cartilage, loose or dense connective tissue. So once you have revised your anatomy, histology, what are the different types of tissues, whether they can grow, whether they have in the different cell cycle, and so what do you require to go ahead with tissue engineering? So what we require is a team, a good team, where you have a cell biologist, a clinician, and a material person with you. Just like any other product development that we talk about in engineering, you need a team, raw materials, design, implementation, verification, validation. The same checklist we have in tissue engineering as well, like quality assurance, maintenance. So, if you need to why do you need tissues? Because some particular tissue has been damaged. So it could be to repair a tissue, it could be to replace a tissue, or it could be to refine the damaged tissue. And what do you plan to inject? It could be cells or tissues. So which means that if you have cells of different sources, whichever have you targeted, if you know the culture techniques, you can put them together. So you can go ahead with cell or tissue replacement. And if you add a matrix to its scaffold, then you are ready for a tissue engineering. So basically, if you want to go ahead with tissue engineering, we need a matrix, a scaffold in which you would grow the cells. You want to target which types of cells you would like to use, and then you would like to add growth factors or the biomolecules which regulate and which stimulate the differentiation of these cells. And so the principles of tissue engineering are the same. For each tissue, you may have to define them. What is the best scaffold? What is the best cell? What is the best molecule? And then finally, these tissues, you may or may not want to go ahead with forming an organ. So these are the basic requirements. Within the scaffold can be a biomaterial, or it can be a decellularized natural tissue. Basically, you're giving them the right niche for them to grow. You could take the help of other external tools like bioreactors or other techniques to go ahead with tissue engineering. But this is a fundamental principle which guides any tissue engineering. What are the different biomaterials that you can use? I'm sure you people are much more familiar with these. There could be nanomaterials, silk, chitin, polymers, metals, so natural substances, modified substances, synthetic materials, ceramics, semiconductors. So this in clinical practice, the dental implants, bone implants, cardiac valves, intraocular lens, prosthesis, contact lens are already being used. So, regarding cells, if you want to deal with cells, human cells or cell, you need to have the regulatory approval. The prerequisite for any cell or tissue therapy is you need to have appropriate regulatory approvals. The cells should proliferate to sufficient quantities, and they should differentiate into desired cell types. They should survive, network, integrate into the host tissues, function appropriately, and should not cause any harm to the recipient. So, which cells are you going to use? As we just talked about, are you going to use adult cells? But if you want them to divide, they should have some proliferative capacity, preferably stem cells or progenitor cells. You could use adult stem cells, which have least of ethical issues. Fetal stem cells have a lot of ethical issues. Embryonic stem cells are again pose a lot of ethical issues. The induced pluripotent stem cells, the Yamanaka factors. So basically, in any adult cell, if you can add the Yamanaka factors, you can convert them into induced pluripotent stem cells. So that is again another area of research. So you can also plan of mesenchymal stem cells, or what are the different scaffolds. So the challenges which you need to address is, are you going to use the allogenic cells or autologous cells, homologous or non-homologous? That means you want to use liver cells for liver tissue regeneration, or you want to use another bone marrow cells to make liver cells. So that's homologous versus non-homologous. You want to use differentiated versus non-differentiated cells. Do you want to expand them or do you don't want to expand them? Would we looking at pure cells or a mixed composition of cells? What could be the route of injection? How many cells? What scaffold to use? Are we making 2D or 3D? How long will this scaffold survive? Do we want it to be for a shorter time or for a longer time? Do we want it to degrade? Don't not to degrade? And once we put it, is it appropriate to get vascularized? Is it would it survive? Would it integrate? And would it show functional recovery? So this is the ultimate purpose for which you need to make tissue engineering. So you have to, even if each of us is not addressing this issue, one of the team members would be looking at this so that we are ready with the appropriate product. So what are the regulatory requirements which I just mentioned to you about? So once you know what tissues to make, read about the tissues, you know what materials to use, how do we go ahead with regulatory requirements? So the institutional review board or institutional ethics committee is the one which has to approve your proposal, which you have to make in a full length proposal, what you intend to do, where do you get the tissues from, whether it is human tissues, how are you going to get them, which organization is doing it, which organization is participating, your collaborators, everyone should agree, you should have informed consent, and all the ICMR and DBT have given excellent guidelines how to prepare a proposal for ethics committee. You need to have the approval of ethics committee. Then institutional committee for stem cells. In case you intend to use stem cells, then you have to have an approval of another committee, which is called institutional committee for stem cell research. If you're using a material, then the biocompatibility testing, toxicity studies. If you're using any engineered substance, then it is biosafety, institutional biosafety has to approve your proposal. Infrastructure, if it is a preclinical or a clinical use, do you have the appropriate CGMP facility, good manufacturing practice facilities to proper. So this is for scaling up. It doesn't come at every stage, depending on where it is applicable. And for clinical trials, you need to register at Clinical Trial Registry of India. So the approach to tissue engineering. The next step, once you have your regulatory approval, you have to review the progress of the models. So let's review what has been happening across different sectors and with different tissues. And this is, again, review of literature from published material, not my own, and how we can do a structure-function correlationship. So this is a review of what's happening in cardiology, diabetes, neural, and I would be looking at few things. If you want to talk about heart, so somebody's heart fails, we repair it by the blood vessels, if they are blocked, the block is removed by bypass or angioplasty. But if the whole heart fails, and if the patient is ready for a cardiac transplantation, we don't have adequate donors. So if we want to synthesize the heart, it should have adequate chambers, it should have appropriate flow of blood, and it should be able to separate the oxygenated from deoxygenated blood. It should function lifelong, and it should pump to the rest of the body with adequate pressure so that every organ of the body is adequately getting blood supply, and it should be biocompatible. So for heart engineering, they have used xenografts, porcine. However, there are a lot of issues, and therefore, to the best of literature search, the best of the human biocompatible heart is not yet ready. It's human donor transplantation which replaces the heart. Blood vessels. How do we prepare a blood vessel? So blood vessel is a tubular structure with multiple ports, depending on whether it is a capillary, whether it is a medium-sized artery. So it has a muscular port, it has an elastic port, it has an outer port, and an inner port of endothelial cells. So in case you want to make a tubular structure which mimics a vessel, you need to line them by endothelial cells. And attempts are being made at different centers to make a blood vessel which can be biocompatible. and probably can be even lined by autologous cells. These are attempts to make synthetic and biosynthetic cardiac valves. So the reason why you need to have endothelial lining is if the blood comes in contact with any foreign material in the body, it will clot. So if and to prevent that, they need to be lined by endothelial cells, and the patient also has to receive what is called as clot preventing drugs. How do we attempt for pancreas? It's an endocrine tissue, so therefore the difference between exocrine and endocrine is the endocrine hormones that are directly secreted into the blood, and that is the reason why they are richly supplied by blood vessels. So we need to decide, are we just going to supply insulin or supply the cells which secrete insulin? Insulin, you can do it by injections or insulin pump, which responds to blood glucose levels. But if you want to plan for pancreas or tissue, pancreatic tissue, then what again? Think of what sources are you going to use? Are you going to do research using embryonic stem cells, mesenchymal stem cells, fetal cells, autologous cells? Depending on your team's area of expertise, one would pick up what type of cells. If it is, then you need to look at how are you going to hold the cells, how are you going to put them into the patient. So you need scaffolds, or how do you hold the cells? How do you pack them up and put into the body? And there is a one good concept of what is a promising concept of called as a missile, or the cells are packed in semi-permeable membranes where they respond to glucose levels, but the immune cells don't disturb them. So the life, how do you replace the cells? And you should have a strategy for replacement, and it should be biocompatible. Skin engineering, again, the progress has been made in under different teams. It should be autologous skin can heal by itself, but if you want, you can expand it in the lab. But if it is too much of a deficit, then you have to pick up cells and expand them, make a skin. Do you want just the epidermis, or do you want the dermis as well? Dermis means it should have fat in extra. So the question of what we need to make should be crystal clear: cells or just the epidermis, or the entire epidermis with dermis. So, and if you whatever the tissues you're making, will it function? Like example, if it is a whole skin, will it withstand the temperature, exposure, moisture, infection? It is biocompatible. These are the questions that we need to ask ourselves. What's happening in the area of neural? That is the most challenging area. Even if we make cells which look like neurons, will they integrate? Will they network, evoke action potential? It's like having different computers in the lab without internet. You can't send messages to each other. Central nervous system is something like that. The neural, there have been attempts to expand the cut ends of a nerve by providing a neural tube. And this is, again, review of literature. Attempts have been made to damaged kidney. We all are familiar that the patient will undergo dialysis. Tissue engineering of liver throws different challenges because it's like a factory and a complex organ which has hepatocytes, which has endothelial cells, which has bile ducts, canals, and so it's basically secretory, a factory which has a lot of metabolic activity, and then a collecting or excretory system. So all these to be engineered is a huge challenge. And what is the scaffold? How do we have these 3D mimics? So these are the challenges which one faces in liver regeneration, liver tissue engineering. The tooth, this is something very exciting, not my area, but this is to complete the review of literature. There have been attempts to collect the milk tooth stem cells because they have stem cells from dental pulp. They have been, they could be expanded, and there are neurotrophic factors which are there, and different odontogenic tissues can potentially be created by using the cells and matrix. Another very promising clinical trial, which was published in Lancet a decade ago, was what is called as a tracheal tissue engineering. So a cadaveric trachea was taken, decellularized, and injected with patient's own autologous cells, and it was transplanted. This was an anecdotal successful case of tracheal tissue engineering. So the concept of decellularized tissue is something like we all did as children. You take a leaf, put it in between our book diary, and wait for it to get dried and see the skeleton. So a decellularized tissue is similar to the dry leaf. If you powder the leaf, it becomes an extracellular matrix. But it's intact, it's similar to decellularized. So you have two options. If you want to make a liver, like for example, you can use a scaffold which mimics a liver and put cells, or you can use a decellularized liver and inject cells. So these are an overview, some of the reviews which have been published. So what is extracellular matrix? Extracellular matrix is similar to the cement which you use in construction. So this is a binding material which has both cells, which has fibers, and which allows a lot of blood vessels, and it is not a quiet inert substance. It is quite an active, and it modulates with time and in the different conditions. So how do we progress? So now here is some examples I'm giving you with my personal experience related to ophthalmology, where we progress from basic research to clinical research. So again, where do we start? So I'm asking myself the same question. And what cells would I use? Do I have enough information about the cells? How do I expand the cells? Do I do it in vivo? Do I do ex vivo? Do I do I have adequate faculty, facilities, funding, and freedom to do? So these are some of the publications which you can find by Googling Vemuganti GK. So the ocular surface is the picture as is shown here. So the white part of it is covered by conjunctiva. The transparent part is the cornea, through which you can see the iris, and the center uh, area is called the pupil. So this ocular surface is, if you see histologically, it is a conjunctival epithelium which ends in limbus. This round thing is called, what is similar to a clock, is called the limbus. The zone between the white and the transparent part of the black part of the eye is called the limbus. So this is the limbus histologically, and it continues with the cornea, and this is the transparent cornea. So what is unique about this? The unique things about cornea is it is transparent, it does not have blood vessels, whereas the white part, which is lined by columnar cells, goblet cells, with it has loose connective tissue underneath, is not transparent, and it has blood vessels. The junction, we know that the skin cells grow from the bottom layer to the top layer. So, and it should have appropriate niche to hold the stem cells. But if the cornea doesn't have blood vessels, how will it hold the niche or the stem cells? So, nature has provided the stem cells for corneal epithelium in the limbus, that is this round part of the this round circular thing which divides the transparent part from the white part is called the limbus, and that has the limbal stem cells which repopulate the corneal epithelium. So what happens? Why did we have to look at this? We had to look at this because some patients have chemical injuries, because of which the limbus is damaged. If the limbus is damaged, the stem cells which repopulate the corneal epithelium are lost. So therefore, the surrounding white part of, that is the conjunctiva, will overgrow the cornea and will lead to vascularization, fibrosis, and loss of vision. So even if you remove this and do corneal transplantation, it is not adequate because the stem cells, that is the limbus, has been damaged. So what is the solution for this? This again, it is something not which we did not invent. There was already a publication in Lancet by the Grazilia Pellegrini group. What they did was, so what the surgeons doing to treat these conditions of limbal stem cell deficiency? They would take few clock hours of tissue and transplant it back to the eye. But instead of that, if you know that they have stem cells, the bioengineering of the limbal tissue means you take 2 millimeters of the limbal tissue, take it to the lab, use an appropriate scaffold, grow into a sheet of epithelium, and transplant it back to the damaged eye. So this was one of the there were two studies which were published, and following this cue, so we tried to do this study using cadaveric tissues. We prepared the appropriate media, we prepared the appropriate human amniotic membrane, we modified the amniotic membrane to make explant cultures. So, what was the strategy to do this experiment? The strategy of doing this experiment was, instead of using the uh, dishes which are commercially available, we made an indigenous explant culture system where we took a glass slide, we took amniotic membrane as a substrate, and we made explants of the limbal tissue, and we watched these cells grow around the explants, making a sheet of epithelium. Do we have to use amniotic membrane only? No, you can use amniotic membrane, you can use fibrin. So these are the different scaffolds which can be used to grow these cells. So these scaffolds can be collagen scaffolds, and this was one of the works that we did, where we used collagen scaffold, which is transparent and which mimics the normal cornea-like structure, which is transparent, on which you can grow the cells. So once you have grown the cells, what do? You can go for cell therapy. If you have the checklist ready, that means you have sufficient cells, desired cells, you can go to the clinical scenario and transplant these cells. So this was already published, where the protocol requires that you can take a tissue, transport it, take to the lab, expand them, characterize them, and transplant it back to the patients, where you can transplant it. And if the patient sees fine, it's good. If it doesn't see, then you can the patient can undergo corneal transplantation. And if just the cornea or the limbal tissue is not adequate, then you can do what is called as a co-culture. You can put two different types of epithelium. This is another modification of tissue engineering, where one type of epithelium can be put outside, one type of epithelium can be put inside, and you can make a ring barrier or whatever shape barrier you want and segregate them on one single membrane. The proof of survival came from the clinical evidence, where the patients have been followed up for more than 10 years and they have been doing well with more than 60 to 70% clinical success. If we want to make a 3D cornea, what we have made is only corneal epithelium, but if you want to make a 3D cornea, we need to use the corneal stroma, we need to inject the cells, and we need to add appropriate growth factors. Can we make retina? That's a different challenge. Retina is an extension of central nervous system which has 10 layers of cells. And this has both neural cells, glial cells, and other cells, and photoreceptors. There have been attempts by previous workers where they have tried to inject fetal cells, but because of ethical issues, it could not be continued. People are using induced pluripotent stem cells to make retina, and all are in bench phase. None of them are in clinical phase. So if you have to make the retina, what are the questions that you will ask yourself? Same checklist. Which type of cells? Will they grow in sufficient quantities? Will you have the 3 types of cells? Will you have photoreceptors, glial cells, neurons? What is the media that you would use? Would you make 2D? Would you make 3D structures? If you transplant it, will it integrate? Will it survive? Will it be accepted? Will it be functional? Will it cause no harm? Will it survive, integrate? And what is the way, how it is functioning? Is it differentiating or is it integrating? So this is the checklist one has to go through before attempting for retina. And another basic question is, all that looks like neurons, will they function like neurons, is a basic question. And this can be addressed by doing electrophysiologic studies or niche-directed studies. So this is another study which was done by our group to attempt to treat dry eye. So that is the lacrimal gland insufficiency. So we tried to make the lacrimal gland. This is the summary of the work already published. The human lacrimal gland cells could be expanded. They could be made into sheets of epithelium. They could be made into 2D, 3D structures. They could be characterized, and they could be made as lacrispheres. Yet, We don't have an appropriate scaffold to make an entire lacrimal gland. We are not ready for clinical transplantation. So the tissue engineering component of it is still incomplete because only the cells have been grown. One promising thing about this is when they were grown, the tear cells could secrete tear substances into the media, which gives us a promise that this can potentially be used for therapeutic applications. So this is what is called as an evidence. This is pyramid which shows the hierarchy of weightage for different types of studies. So the evidence-based pyramid shows that the in vitro, the test tube research, that is most of the studies are only in vitro or animals, or if they are opinions or they are review articles, and this is where most of the research is. And if you can do one or two cases, then it becomes a case report, then case series, then case control studies with a control group, cohort studies, and with the randomized controlled double blind studies, and then a systematic review of all these studies. So this is what is called as a pyramid. So the evidence becomes much more stronger. The height of evidence comes from systemic reviews or meta-analysis of all the studies done. So we are far, far from that point, except in few areas. So our tissue engineering for potential clinical application is still at the bottom of the pyramid. The future direction of tissue engineering is exploring the possibility of microcapsules with semi-permeable membranes, looking at new scaffolds, biomaterials with growth factors, and attempting to do live imaging of the cells in vitro as well as in vivo, so that we see where the cells are growing in this tissue, in the engineered tissue. How do we track these cells? How do we follow up their survival? Can we use bioreactors? Can we use gravity to make 2D and 3D structures? And what is the best way to deliver the cells to the target tissues? So these are the potential new areas which many of us can explore into. So basically, tissue engineering is a very exciting field of science which requires a complementary teamwork, and it would contain cell biologists, clinicians, biomedical engineers, material sciences, etc. And that's when all of us put together can make attempt to simulate nature and to make a tissue which can potentially be transplanted to or put to use to humans. And I would like to acknowledge all my clinical colleagues, my collaborators, my scholars, my project fellows, technical staff, all the funding agencies who have supported the work that I just showed in the last few slides, and the institutes where I have worked. Thank you very much. I'd be happy to take any questions.
Well, it's time to thank Professor Geetha Vemuganti garu for delivering such an excellent and informative session. Thank you, ma'am, firstly for accepting our invitation and being a part of this webinar cum FDP series. And I would like to thank from the bottom of my heart for such a wonderful session and for enlightening the audience with your profound knowledge, ma'am. Ma'am, your choice of words are nothing but words of wisdom, and these words of wisdom would echo in our minds for a very long time to come, ma'am. Thank you very much, ma'am. We are blessed indeed to have got the opportunity to listen to you today, ma'am. Thank you very much.